Welcome to part 1 of our video series on translation, also known as protein synthesis. In this process, the genetic code carried by mRNA is decoded to build a polypeptide chain. In this part, I will explain the location and requirement of translation, including ribosomes, messenger RNA, and tRNA and their key features. Additionally, the charging of tRNA, proofreading mechanisms, and differences between prokaryotic and eukaryotic translation will be discussed. Major translation process occur in cytoplasm and we will focus on that. However, there are few proteins which are synthesized in mitochondria as well. There are three broad requirements of translation. The first one is ribosomes, which provide coordination sites for the translation. Second is messenger RNA, which serves as a template, providing information for a polypeptide chain. And the third one is tRNA, an important molecule that transports amino acid to the site of translation. In addition, it also reads and decodes the genetic information and codes it in the messenger RNA. So in the coming slide, I will discuss the key features of these requirements one by one. So let us begin with ribosome, which serves as a micro machine for manufacturing protein. It consisted of two subunits, the large one which is catalytic in function and the smaller one which is a decoder or reader arm. Basic structure of ribosome is same both in bacteria and eukaryote as well as in mitochondria, consisting of two subunits, the large and the small subunit. In bacteria, 50S is the large one and 30S is small one. Together they make ribosome, which is 70S. In eukaryote, the bigger and the small one together makes 80S ribosome in eukaryote. S here is indicative of size, which is Swedberg unit. It is also called as sedimentation coefficient. It means the size and the density of eukaryotic ribosome is higher than the bacterial ribosome. So these subunits remain separated. They assemble only in response to translation signal, and the assembled ribosome participate in the translation, not the separated units. This assembly requires role of a special protein known as initiation factor, which we are going to discuss later in this video. There are three important sites in ribosome where coordination happens during translation. Site A, two reactions happens. It accepts amino acid tRNA and the peptide bond is formed here. So this is known as elongator site or elongator tRNA. The second is P site, two reaction again here. It accepts initiator tRNA, which is a special kind of tRNA to deliver methionine in every protein. And the second is the peptidal bond tRNA, which is formed at A site, is accepted here also. Now E site accept the uncharged tRNA, which is formed as a result of formation of peptidal tRNA at A site. From E site, this uncharged tRNA is thrown out of site, and there is another site which is known as protein exit site, through which the polypeptide emerges out during translation. The next requirement is transport RNA or tRNA. This is a small RNA molecule having less than 100 nucleotides. It has unusual bases and modified bases. It also has intra-strand complementarity, thereby forming cloverleaf-like structure, which is shown here. There are more than 22 different types of tRNA to ensure that at least there is one tRNA for each amino acid. There are two important sites which you have to remember with respect to translation. The first one is acceptor arm. It has acceptor arm where amino acid is loaded and carried to the site of translation. And the second is very important site, which is a reader arm known as anticodon arm. This is to read the genetic code encoded in the form of messenger RNA. The first amino acid in every protein, both in prokaryote and eukaryote, is methionine, which is delivered by a special tRNA known as initiator tRNA. However, the internal methionine residues are delivered by a different tRNA known as elongator tRNA. Please note that the structure of these two tRNA is different. So the first amino acid methionine is delivered in each and every protein by a special tRNA known as initiator tRNA, which is slightly different in formulated methionine in prokaryote. It is slightly different than the eukaryote, which is pure methionine tRNA. So this is a difference between prokaryote and
eukaryote. The another tRNA for methionine is elongated tRNA that specifically transport the internal methionine residue in a polypeptide chain. The structure of these two tRNA is different as well.
To synthesize a unique and correct polypeptide chain, an amino acid should have a unique tRNA. I have shown here three tRNA. One is for alanine, the another one is valine, and the third one is for methionine. They must bind to their respective amino acids. If there is any mismatch, that is rejected by the enzyme known as amino acyl tRNA synthetase.
So the question is, why is tRNA needed? There are two reasons. First is activation of amino acid. Under normal physiological conditions, a peptide bond cannot be formed between two amino acids because this is not thermodynamically feasible. Second is amino acids cannot read genetic code and specify a unique amino acid sequence of polypeptide chain. Therefore, you need tRNA to activate amino acid and to read the genetic code in the messenger RNA. The question is, how is an amino acid activated? So amino acids are activated by their binding to their corresponding tRNA, which is known as charging of tRNA. This reaction is also known as amino acylation of tRNA. This reaction is catalyzed by an enzyme called amino acyl tRNA synthetases, which activate the carboxylic group of amino acid, forming the highly reactive product amino acyl tRNA. These are the precursors of translation. So amino acids are delivered in the form of AAtRNA to the site of translation. These enzymes are highly special because they have a proofreading activity also. The charging of tRNA is an energy dependent process, and therefore you should know the energetics of the reaction. It occurs in two steps. In step 1, amino acid plus ATP are converted into amino acyl AMP, which is high energy intermediate, plus pyrophosphate, which is ultimately converted into 2 inorganic phosphate, thus driving the reaction in a forward direction. In step 2, amino acyl group is transferred from AMP to tRNA by the enzyme, forming AAtRNA plus AMP. Thus, the overall reaction is amino acid plus ATP plus tRNA, producing AAtRNA plus AMP plus 2 inorganic phosphate. Thus, there is consumption of equivalent of 2 ATP molecules per amino acid. The enzymes are highly precise as they have proofreading activity as well. So these charging enzymes are highly precise and they do so by selecting the correct tRNA for a given amino acid. Therefore, we should discuss the criteria utilized by these enzymes to select the correct pair of amino acid and its tRNA. So any one of the three criteria listed here is utilized by enzyme to select the correct tRNA. In some cases it is the acceptor arm which is utilized. In other cases it is 370 Cg base pair, and in other cases it is the anticodon arm which is utilized to select the correct tRNA for a given amino acid. For selecting a correct amino acid for a cognate tRNA, enzyme utilizes three criteria including the size of amino acid, thermodynamic constants or binding energy, and the third one is hydrophilicity or of the amino acid. When the size is distinctly different, there is no problem, but problem arises when the size is similar of the two amino acids. In those cases, the thermodynamic constant or the binding energy is also same, therefore enzyme makes more error. In such cases, this is the hydrophilicity which is used to distinguish between right and wrong amino acid. In spite of being highly precise in selecting the amino acid and tRNA pair, a synthetase enzyme makes errors. The normal error rate is 1 out of every 10,000 amino acyl tRNA. This is increased significantly when enzyme is dealing with similar sized amino acid. The error is enormously increased to 1 out of 100 amino acyl tRNA. This must be removed, otherwise you will see appearance of substitution in the protein being produced. The incorrect amino acyl tRNA is removed by proofreading activity which is known as double C mechanism. Before I discuss double C mechanism, I would like to explain how is an incorrect amino acyl tRNA formed. So let us discuss how incorrect amino acid tRNA is formed, taking example of valine and similar sized threonine. Valine is hydrophobic because it has a CH3 group, whereas threonine has methyl hydroxymethyl, and this is hydrophilic amino acid. The valine must bind to its corresponding tRNA for valine, and majority of the product, about 99% of the product is correct, which is valine going with tRNA for valine, and this participates in the translation. About 1% of the product, because of the similar size, threonine is bound to the incorrect tRNA and forming incorrect product threonine with tRNA for valine. This is a wrong product. This must be removed by a mechanism called double sieve mechanism. So what is the double sieve mechanism? I will discuss in the next slide. So what is double sieve mechanism? Double sieve means there are two sites in the enzyme, which is shown here by the rectangle with the green background. There are two sites in the enzyme, catalytic site and the hydrophilic proofreading site. At catalytic site, the product is formed, which is here valine with tRNA for valine, and this is the correct pair. This is the correct product, and you have learned that this is hydrophobic, therefore it cannot enter the proofreading site, which is hydrophilic, and it is protected and participates in the translation. So no entry into the proofreading site is a certification of authenticity of the product. The incorrect product is also formed on the catalytic site, which is threonine going with tRNA for valine. This is a mismatch, and therefore this is a wrong product, and you have learned that this is hydrophilic. So being hydrophilic, it easily enters the proofreading site, where it is degraded into its component amino acid threonine and tRNA, which are then recycled. So this is about double C mechanism. So the last requirement is messenger RNA or mRNA. I will discuss structural features of the messenger RNA which are relevant to translation both in prokaryote and eukaryote separately. So prokaryotic messenger RNA has three distinct regions: 5' UTR, 3' UTR, these are regulatory regions, and ORF, or this is the open reading frame, which has information for a polypeptide chain. It is ORF start with AUG, which always codes for methionine, and it ends with one of the three stop codons listed here. So these are also called as terminators. In the 5' UTR, there is a purine-rich region, which is a site for binding of ribosome. Now it is noteworthy that the ribosome is assembled right on the start codon in prokaryote, which is not the case in eukaryote. Eukaryotic messenger RNA also has three regions: 5' UTR, ORF, and 3' UTR. These 3 and 5' UTR, these are regulatory in function. ORF is same as in prokaryote, having information for a polypeptide chain. It has the first AUG, which codes for methionine, and also the same stop codon. One of the three is good enough to stop the translation. There are some differences. 3' UTR has poly A tail, which is not the case in bacteria. 5' UTR has capside, which is absolute requirement of the ribosomal binding, and this is not the case in bacteria as well. So ribosome is quite far away from AUG start codon, therefore it scans for the correct AUG. What I mean is that AUG present in a special consensus sequence, which is known as Kozak sequence. So for that it has to scan, and that scanning is an energy dependent process. Thank you very much for watching this video. Please like it, share it, and subscribe it. Until next time, you take care. Bye bye.